In this video, we're going to look at kidney histology. So you can see here, there are two views, one of the kidney's cortex, which is the outer part of it, and then one of the kidney's medulla. So what we can see in the cortex are several glomeruli, which is the ball of capillaries that has come from the afferent arteriole, and that's where filtration of plasma will start to happen. Notice that we don't see the glomeruli in the medulla view because that's mostly renal tubules and connective tissue. So the renal tubules are all of these surrounded by, in this case, simple cuboidal epithelium. There's several of them. And then if we look over here in the cortex, we can also see some of the tubules. And then over here on the outside, that's the kidney's capsule, that connective tissue layer that helps to keep all the cells together and keep the organ intact. So let's switch to a higher magnification so we can look at the glomeruli in some more detail. Now we're looking at a highly magnified view, a total magnification of 400x, of two different glomeruli from the same slide. So we're looking at two and they're positioned slightly differently so that you can see the common features between them. You can see that the ascending loop of Henle has been marked for both glomeruli, or both structures. The glomerulus itself is here and here. And then next to it, we see two cells that are critical in helping this kidney determine the rate of filtration and help to regulate blood pressure. The cells here that are closest to the glomerulus are actually in the walls of the afferent arteriole, and those are granular cells. So these cells serve two important functions. One is that they serve as baroreceptors. So as they're in the walls of the afferent arteriole, they're trying to detect enough stretch in the walls of that arteriole to say there's a good amount of blood coming into the glomerulus and that we should have adequate GFR, or glomerular filtration rate. The other thing that these cells do is they secrete renin if they detect that that pressure is too low, because if the pressure is too low, then filtration rate will be too low as well. Renin, if you recall, is used to convert angiotensinogen to angiotensin 1. And then lastly, we'll look at the macula densa cells. So you can see that these cells are actually part of the wall of this loop of Henle, and their job is to serve as chemoreceptors. So they're monitoring filtrate that's about halfway through the nephron, maybe about 2/3 of the way through the nephron. And if they detect problems in sodium content that would indicate we need to elevate blood pressure or GFR, they tell the granular cells to release renin so that we can do that. We can create that cascade of events that will actually make that happen. The granular cells, the macula densa cells, and the extraglomerular mesangial cells, which help those two communicate, are collectively termed the juxtaglomerular apparatus or the juxtaglomerular complex.